Since water is the most abundant molecule in the body, anything that concerns water is going to be important. And although water can move in a variety of ways, osmosis is really what operates at the cell level because it's how water moves across a membrane. The way that happens can be a little confusing, so let's explore that in this video. Well, osmosis is another type of membrane transport, but this time, instead of looking at how these particles are moving, we're looking at the other 99+% of the particles there. The water particles, all the blue stuff. So we know that these particles would want to spread out from high to low concentration. We're not concerned about that with osmosis. We're concerned about what happens to the blue stuff, most of this stuff here. It's going to move in this direction. And understanding why is a little confusing, but let's start with the idea that osmosis is a passive process just like simple and facilitated diffusion. In fact, it's kind of a combination of those two things. So on either side of a membrane, you have water, and lots of it. And really the difference between the water concentration on either side is not terribly different. Usually it's these solutes, the stuff dissolved in water, that's more different in a noticeable way. But let's just talk about how water gets across the membrane. It's actually two ways. One, even though water is polar, some of it can sneak through the membrane. Not a ton, but a bit. The cell can also make membrane proteins, and these are called aquaporins. It's a name that makes a lot of sense. Aqua for water, and pores. They're water pores. The more of these aquaporins that the cell makes and puts in the membrane, the less resistance there is to water moving. More aquaporins, more water movement. So cells have some amount of control over how much water comes in and out. So the big question is, what drives the water movement? With other types of passive transport, we talked about diffusion, things moving from a high to a low concentration. But what about with water? I said these concentrations were not terribly different. It's usually the solute concentrations. And there's a lot of stuff dissolved in the watery fluids within our bodies, whether it's inside or outside the cell. There's a lot of different electrolytes and ions. There's proteins, there's sugars. All those are solutes. And so it doesn't matter what it is, but these are going to drive water movement. And kind of a nice rule of thumb is water follows salt. Kind of keep that phrase in mind and don't get too married to the idea that these are salt particles, but any sort of solute, then you got it. So in this case, there are more solute particles over here, which is why I keep drawing water moving that direction. Water is going to move this direction until the solute concentrations on either side balance out. Because eventually there's going to be a lot more water over here, and these particles are going to become diluted until they eventually match the solute concentration over here. That's it. There's a couple different ways I can see this playing out in your mind. One is that water itself moves from a high concentration to a low concentration. Now the difference here is negligible. This is 99.999% water, and this is 99.998% water. But there's still a difference in concentration, so water is going to move down its gradient. The other way, the flip way of looking at this, is from the solute perspective. We could look at water as a helper. Water is trying to move out. Since this solute cannot come across this membrane, water looks at that solute and says, oh, poor guy, let me help you out. Let me move across the membrane so that you can spread out like you want to. And because normally this solute would move over here until concentrations were equal. Well, water can accomplish the same thing. If water moves this way, it will do so until the concentrations of that solute are equal. Whichever way makes the most sense to you is the way I would think about it. Let's look at a couple slides to drive the point home.
Now, osmosis can seem really simple when presented that way, but it gets more complicated when you put it in a situation. This is a weird one. So we have this U-shaped tube, and the key thing here is we have this semi-permeable membrane. We also have solutes. Doesn't matter what it is. Let's just say it's salt. And there's more of them over here on the right than there are over on the left. And so you can see that there's more water on the left and less water on the right. And in this case, these solutes can't move through the membrane, but water can. So the question is, which way will water move? Well, it moves to the right, and you notice that it's done so until the concentration on the left equals the concentration of solutes and water on the right. The challenging part about this is, I think, to overcome the mental hurdle that water just sort of levitated against gravity to equalize solute concentrations. It's one thing to say just water moves from left to right, but another one to say that it moves up against gravity. Shows you that it's a pretty powerful force. We can see that in plants as well. So if the environment outside of a plant cell is really salty, then water moves out. And since that cell wall sort of stays in place, this cell inside shrinks away from the cell walls. If the solute concentration outside the cell is the same as the solute concentration inside the cell, then the balance of movement of water in and out equal one another. But if there's a lot more solutes inside the cell than outside the cell, more water moves into the cell, and in this case, into the vacuole of a plant cell, creating this turgor pressure, causing it to push against the cell walls and the plant stand upright.
Well, those examples on the plant, you could see what a difference it made to the cells. And that holds up for the plant as well. So this is a plant, poor thing, that does not have enough water in its cells. Couldn't maintain that pressure in its lymph. This one, all that water pressure inside the cells is holding it up, at least mostly. You probably saw some of the terms on there, like hypertonic. So this idea of tonicity is a really tough one, but important even when it comes to us, especially with intravenous fluids. So a quick introduction and then a slide. So tonicity always uses the cell as a reference. So it's just basically saying whatever salt level is in here is essentially zero. If we have less salt over here, well, that's a negative, right? It would be less salty than inside the cell, or less solutes. If we have more solutes, well, that would be a positive. And so it's just saying we have more solutes out here. And that then determines where the water goes. Remember, water follows salt. So in this case, there's more salt in here, so the water would come in. In this case, it's going to follow the salts out. Really different results. Let's take a look at that with red blood cells.
All right, so here we have three very different looking red blood cells. These in the middle, this kind of weird disc-shaped ones, that's how red blood cells are supposed to look. Using the cell as a reference, and the inside of the cell, remember, as sort of zero, the sort of amount of saltiness it should be, then over here to the left, that has more solutes in the solution. And the one over here, the hypotonic, has less solutes in solution. So what is sort of normal? It's about 0.9% sodium chloride. This is also referred to as normal saline. This is often a term you'll hear in medical situations, whether it's an intravenous fluid or just fluid that you're putting in a wound or something like that. You've probably encountered this with eye drops. So normal saline is what you get in eye drops, and they feel good in your eyes. But if you get super salty water in your eyes, hypertonic, it hurts. And the same is true of pure water on the other side. It also is somewhat painful or at least uncomfortable feeling. So what's happening here in terms of osmosis? Well, obviously each of these cells is surrounded by a membrane, and in this case, the amount of salt inside and outside is balanced, and so is the movement of water. And so the cells stay just right. Over here to the left, the hypertonic solution, there's lots and lots of salt on the outside, which is drawing the water out, and the cell shrinks in because animal cell walls are flexible, and so that just shrivels up. You can imagine that would cause problems in the bloodstream if you were to put really high salt concentrations in there. Now on the right over here, we have pure water, and you think this would be great. Just put in pure water, the person needs to be hydrated. But the problem is the inside of those cells still has salt. And if you have a bunch of pure water, then a lot of water is going to come in here, even potentially bursting that cell open. It can create so much pressure inside this cell. Without a cell wall to protect against that pressure, the cell can literally blow up.
Well, there are a lot of practical applications to osmosis, as you might imagine, since water is so important to life. Not only with plants, by the way, another way that you can get plants to wilt is by over-fertilizing them. Because fertilizer is mostly salt. When you put that in the soil, it sucks water right out. Also, the intravenous fluids. We're bathing our red blood cells in whatever fluid goes in. So it's important that that's isotonic or really close to it. Some other applications would be things like sweat, right? It's a normal body function. But why does sweat taste salty? When it runs down your forehead and you taste it, it tastes salty. Because the body is pushing salt into those little tubes. Water follows it, and that creates water pressure that pushes water right on out. And then a really important one, for people with high blood pressure, they're told to avoid sodium. Sodium is a really big driver of osmosis. Water really seems attracted to salt. And so, people with high blood pressure, if they have too much salt in their bloodstream, that attracts water into those blood vessels, which don't really grow that much, and so it creates bigger volume, pushing against those vessel walls. That's what high blood pressure is. So there's a good introduction to osmosis. There's a lot more applications we could make with it, and a lot more detail we could go into. I'm sure you'll get into it in the future more. If you keep the idea of water follows salt in mind, then I think you'll usually be able to predict where water goes. An important concept to be sure. Until next time, keep appreciating life, even little things like osmosis.